Hello to all my dear students. I am Deepen Singh. Welcome to all of you in our school SJEC e-learning YouTube channel. So student, as we know, we are discussing our chapter 8 circulatory system. And in this chapter we are already discussed the components of circulatory system, which are body fluid such as blood, lymph and tissue fluid, blood vessels and lymph vessels, and the last but important component, heart, to pump the blood through blood vessels. And in this topic we already discussed in detail blood and its composition, such as plasma and blood capsules. But in today's video we are going to discuss in detail WBCs. WBCs, or white blood capsules, also known as leukocytes. White blood cells or capsules are nucleated, irregular amoeboid cells. The number varies from 5000 to 9000 per ml of blood. And if we talk about structure, then WBCs are colorless. That's why the name is WBCs, means white blood cells, due to the absence of color pigment. These cells are rounded or irregular shape, and they move like amoeba and may even come out of the capillaries. And this phenomenon, to move like amoeba and came out of the capillaries, this phenomenon is known as dipedesis. WBCs survive for 12 hours to 12 days, means it depends on the working, means there are some types of cells which may alive 12 hours and some are alive for 12 days. If we talk about the neutrophil, these cells are survive only for few hours, and about 125 billion neutrophils are produced each day. And the worn out cells, damaged cells, waste cells are destroyed in the liver and spleen. And this is the important fact about WBCs, that the worn out cells are destroyed in the liver and spleen. So mark it important. Another process which is relevant to WBCs are leukopoiesis, and the process of WBC formation is known as leukopoiesis. The new WBCs are produced in bone marrow, lymph gland, spleen and thymus. And if we talk about types of leukocytes, then the leukocytes are of two types: granulocyte and agranulocyte, in which granulocytes again divided into three parts: neutrophils, basophil, acidophils, while agranulocyte divided into only two types: lymphocytes and monocytes. Let's see what are granulocyte and agranulocyte. First of all, we talk about granulocyte. The cytoplasm of granulocytes contain granules and an irregular and many-lobed nucleus, means more than one nucleus is present, polymorphic. Based on the nature of granules, granulocytes are of three types. First of all, eosinophils, means acidophils. They are about to 1 to 3% of total WBCs or leukocyte, and the number increases during infection and allergic reaction. Means if a person got some kind of injury or infection, then the number of acidophil increases continuously. and their nucleus is bilobed, as we can also see in the diagram, and the cytoplasmic granules are large and alkaline, means the granule present in cytoplasm are large and alkaline in nature. These granules are stained with acidic dyes such as eosin, and therefore they are called eosinophil or acidophil. The granule contain toxic chemicals which kill large parasites and destroy bacterial toxins and promote tissue inflammation. Clear? Let's move on. Next, basophils. These are about 0.5 to 1% of the total leukocyte, and their nucleus is three-lobed, and cytoplasm has acidic granules. These granules are stained with basic dyes, therefore are called basophils. The cytoplasm of basophil contain heparin and histamine. What are they? First of all, heparin. It is an anticoagulant, means it prevents blood clotting inside the blood vessels. And histamine, it is an inflammatory agent, means it released in allergic response and in injured tissue to dilate blood vessels. Last is a neutrophil. It is the last part of granulocyte and most numerous, forming about 70% of total leukocyte. The nucleus is 3 to 5 lobed, as we can also see in the diagram. Granules are large and are stained with neutral dyes. Therefore, they are called neutrophils. These cells are phagocytic in nature, means neutrophil cells engulf microbes, and this process is known as phagocytosis, means to engulf microbes. They also work as scavengers, means they remove the cell debris. Debris means the waste material of cells, and squeeze out through capillary wall at the site of injury, and this process to squeeze out through capillary known as diapetesis. Our next main component is a granulocyte, means a granular leukocyte. As we can see, the cytoplasm of a granulocyte is without granule but has a large, rounded or kidney shaped nucleus. These are of following two types: lymphocytes and monocytes. Let's see what are lymphocytes. These are the smallest leukocyte. They form 30 to 35% of total leukocyte count, numbering 1800 to 300 per ml of blood. The cells are rounded with a large rounded nucleus and very little cytoplasm. Lymphocytes produce antibodies to provide immunity against infection. Next and last type of WBC is monocyte, and these form the 3 to 7% of total leukocyte. They number just 200 to 700 per ml of blood. Their nucleus is large and kidney shaped, or we can say horseshoe shaped, and if means U shaped. Like neutrophil, monocytes engulf microorganisms and worn out tissue cells. Monocytes are also called wandering cells of the blood. Let's talk about the difference between various types of leukocytes. means the cells found in WBCs. Here is the graphical differentiation between all the types, all the five types. First of all, if we talk about acidophil, the shape is rounded. The size is 10 to 14 mu micron. Nucleus is bilobed. Granules are large, stained with eosin dyes. Function means to detoxify bacterial toxins. Clear? And basophils, these are also rounded in shape, 88 to 14 mu micron in size, three-lobed nucleus. Granules of various size are stained with basic dyes, and the function is to bind numerous antibodies found in the cell. Neutrophils are amoeboid but appear rounded in shape, 9 to 14 mu micron in size, multi-lobed nucleus about 3 to 5 lobes, and the granules are stained with both acidic and basic dye. That's why these cells are neutrophils. Function means phagocytic, cells engulf invading microorganism. Clear? Lymphocyte and monocyte both are rounded in shape. Lymphocyte 6 to 12 mu micron in size, while monocyte 9 to 18 mu micron in size. Nucleus, if we talk about lymphocyte, these are large spherical nucleus, and monocysts consist a kidney shaped or U shaped nucleus. Lymphocyte cytoplasm less and clear, granules few and small. Means if granules are present, these are few in number or small in size, while in monocyte granules are also very few and small in size. Lymphocyte associated with immunity against infection, while the monocytes are phagocytic, means engulf microorganism and work as a scavenger. Clear? What are the functions of WBCs? Leukocytes are described as the mobile force or soldier of the body because they migrate to the site of injury and infection and fight with the invading microbes. They also defend body against microbes and their toxins by forming antibodies, and they work as scavenger, means at the site of injury or infection, neutrophil or monocysts engulf the worn out cells of the body, and phagocytosis means they engulf the microbes and foreign matter or the damaged cells like an amoeba. This is called phagocytosis. Pus formation means in the course of their struggle against invading microorganism, numerous white blood capsules are killed, and these killed accumulate in the infected area together with exuded plasma. These dead leukocytes along with destroyed tissue cell, dead and live microbes and exuded plasma form the pus. Inflammation means swelling caused at the site of injury, and the formation of antibodies means the lymphocyte produce antibodies to kill the germs and neutralize their toxins. Clear? And the last type of blood capsule is blood platelets. As we know, blood platelets are small, oval, and spherical but non-nucleated disc-like structure. They are not cell but fragments of megakaryocyte, which are undifferentiated gained cells in red bone marrow. The function of, if we talk about the function of platelets, they are essential for blood clotting. At the site of injury or cut, blood platelets disintegrate, liberating an enzyme thrombokinase into the plasma to initiating clotting of blood. Now the question is, what is blood clotting? The blood clotting is a function of platelet, or we can say it is the main function of main function of platelet, blood clotting. What happened in blood clotting or coagulation of blood? When a blood vessel is cut, blood comes out of it and quickly changes from a liquid state into a thick jelly like material. This is known as a clot and the process of separation of clot from the plasma. Clear? And what are the steps included in the clotting process? Means the process of clotting involve conversion of solid or soluble blood protein, the fibrinogen, which is already present in plasma, into insoluble fibrous protein. Now the fibrin, which is in the form of fibers, involve the action of number of substances such as prothrombin, thrombokinase, calcium ions and fibrinogen. In first step, platelets release thromboplastin or thrombokinase after the rupturing of cell. Now the manufactured thrombokinase convert prothrombin in the presence of calcium ion into thrombin, and thrombin catalyze the conversion of soluble blood protein fibrinogen into insoluble protein fibrin, and the fibrin at last separate out of plasma as a fine mesh, and the blood capsules get entangled in the mesh and together settle down as a clot or thrombus. Clear? Our next and important topic in this chapter is blood groups and blood transfusion. And in this sequence, the first topic to discuss is ABO blood group. As we know, Karl Landsteiner discovered the human being's blood groups. Means they discovered that glycoproteins on the surface of RBCs called antigens or agglutinogens. Gamma globulins protein in the blood plasma are called antibodies or agglutins. So there are two types of antigen, A and B, and two types of antibodies, antibody A and antibody B, are found in human. Based on the presence or absence of these antigen, human blood falls into following four blood groups. Person with antigen A belong to blood group A. Person with antigen B belong to blood group B. Person with antigen A and antigen B both belong to blood group AB, and person with no antigen or absence of antigen belong to blood group O. And this forms the ABO system of blood groups. Now the topic is antigen and antibody interaction. Means person with one antigen have natural antibodies in their plasma against other antigens, such as the person with antigen A have antibody for antigen B in their plasma, which is represented as antibody B or anti B. And like this way, person with antigen B have antibodies for antigen A. Example, they have antibody A or anti A in their plasma. Person with both antigen A and B do not have any type of antibodies in their blood, while the person with no antigen have both antibodies, anti A and anti B, in their blood. Blood transfusion means the transfer of blood from one person to another person. Means at the time of excessive bleeding because of injury, blood matching is essential before the blood transfusion to avoid incompatibility and blood agglutination. Transfusion fail, especially if large quantity of blood of different blood type is introduced. Either the antibody of recipient blood destroy the RBCs or donor blood, or the antibodies of donor's blood hemolyze the RBCs of recipient blood. In case small quantities of blood are introduced, the blood transfusion is successful only if the plasma of recipient blood doesn't contain antibodies against donor's antigen, or donor's blood has no antigen. It means here is a diagram represented human blood group and their compatibility. If we talk about blood group A, antigen A is present on a red blood cell and the antibodies present in plasma are antibody B. It can donate blood to blood group A and AB, while it receive blood from A or blood group O. In this sequence, AB is known as universal receiver, while blood group O known as universal donor, as it donate all the groups to blood. Means person with blood group AB can receive blood from A, B, AB or O blood group because they have no antibody in their plasma, and therefore person of AB blood group are called universal recipient. Clear? Next and very important factor is RH factor, which is known as rhesus antigen system. Means in addition to A and B antigen, human RBCs also possess RH antigen. Human population is divided into Rh positive and Rh negative person. Rh positive person have Rh antigen, while Rh negative person do not have Rh antigen, and Rh negative person do not have natural antibodies against Rh antigen. When Rh negative blood is given to Rh negative person, the transfusion is successful. Similarly, transfusion of Rh positive blood to Rh positive person has no problem. But when Rh positive blood is given to Rh negative person, antibodies develop in the blood of Rh negative recipient. In this case of second transfusion of Rh positive blood to such a person, clumping of donor's blood cell occurs because Rh antibodies already developed in the recipient's blood which react with Rh antigen. And for the above reason, while determining ABO blood group, presence or absence of Rh factor is also tested and very important in the case of transfusion. Rh person should be given Rh negative blood only. Clear? There is a last compatibility factor for the pregnant lady is Rh incompatibility during pregnancy. And what happened in this case? During pregnancy, the Rh positive fetus in Rh negative mother stimulate the formation of antibodies in the mother's blood. During the first pregnancy, enough antibodies are not produced to harm the fetus. But during the second pregnancy, if the fetus is Rh positive, more antibodies are added in the mother's blood, and a large number of RBCs of the fetus are destroyed due to clumping. This causes hemolysis of blood in fetus, resulting in erythroblastosis fetalis or hemolytic jaundice or anemia in the newborn baby. Clear? So here we complete the today's topic or today's video. Thanks for watching. And student, if you like our way of teaching and hard work. Please subscribe us to learn more and be updated about your syllabus.